Yeah, today I will talk about mechanisms that try to explain how the selectivity of neurons in the cortex, and we try to study these mechanisms using connectomic data to constrain mechanistic models. So when we show, so with current technique, experimentalists can now observe large population of neurons simultaneously. This is an example taken from mouse visual cortex. Here on the left, you have the images being shown to the animal. And on the right, you see population activity. Here, each dot is one neuron. And here, neural activity is recorded with calcium imaging that makes neuron becomes brighter when their activity increases. Okay? So here you see that we can observe thousands of neurons. And what this type of experiments show is that the response patterns are highly specific, to the point that these experiments can show that the animal can distinguish between thousands of different images only looking at the subsets of the one neurons. Okay, so this specificity of neural responses is something that has been studied for a long time in neuroscience, even before these techniques became available. And as I will show in a few slides, there are different models that have been proposed to explain the same phenomena. And these models rely on different assumptions about the connectivity in the circuit. Okay, so because of this fact, what we try to do to develop a different approach in which connectivity is not assumed but is taken from data. And in particular, we rely on newly available connectomic data that I will discuss in a few slides. Okay, so I will tell you more about this in a few slides. Going back to selectivity of neural responses, what is observed in the brain is that neurons show selectivity for the same type of stimulus across different areas. For instance, for visual information processing, selectivity to visual stimuli is observed in the retina, in the thalamus, and across different stages. For instance, this is the ventral stream in macaque. But selectivity is often not redundant, but is gradually transformed across these areas. So along this ventral stream, you have that in V1, neurons are selective for simple features like oriented bars, like this stimuli here. So if you look at the activity of a neuron as a function of the stimulus orientation, neurons in V1 will have a bell-shaped tuning curve with maximum activity at the preferred orientation for that neuron. Then this is in V1, but then if you go higher along the visual hierarchy, neurons become selective to more complex features. Particularly in IT, neurons are selective for objects and faces. Okay. And this selectivity is thought to be important from a computational perspective, but it has been suggested to underlie complex computations such as invariant object recognition. This is the ability to recognize an object regardless of where it is in the field of view, or how it's rotated, or the size of the object. Okay. And there is experimental evidence that this is happening along the ventral stream. For instance, in these experiments, they show that by looking at neural activity only in one of these areas, you can perform invariant object recognition, and this task becomes easier if you look at activity in areas higher along the hierarchy than in V1. Okay, so now I showed you what selectivity of neural response is and why I think it's important to study. So the main question that I focus on is what are the mechanisms that shape this selectivity? So how the network produces this response. And okay, so I will give you an overview of a few existing models, and let me just set up the framework first. So the models can be formulated in this framework, in which there is an input layer that drives the activity of an output layer. And the activity of neurons in this output layer that is made by excited and inhibitor neurons is described by the standard rate dynamics, in which each neuron follows is described by its rate that evolves is driven by the input that the neuron receives from the other cells, passed through a linearity that is the single neuron input-output function. Take a neuron here, it will receive three main types of input: a feed-forward input from the input layer, a recurrent input from the other excitatory cells in the network, and an inhibitory input from the inhibitory cells in the network. And these are these three inputs here. And each one of them is obtained through a weighted sum of the presynaptic activity, so the activity of the cell that projects that neuron, weighted by a connectivity matrix that represents the strength of connection between neurons. Okay. And what is important in what follows is first the average size of this connectivity matrix, that is synaptic activity, and the number of inputs, so how many terms this sum have, that is this k variable, that is the degree of the neuron. This in quartet is typically a large number. Okay, so with this setup, then when it is understanding when this neuron can show selectivity. And I want to make a brief argument that shows why this is not trivial to obtain. Yeah. So let's start from the assumption that neurons in this layer show selectivity. That is, we have the fire rate that depends on the stimulus theta, can be the orientation of the grating or the list of pixels in an image. Okay. The stimulus changes, the fire rate in the input layer changes by quantity order 1. We assume that. And we ask under what condition neurons in this layer are selective. Okay. And so one way to answer that question is to look at the selectivity that the neurons receive. Okay, so this feed-forward input here. So if we assume that the projection from this layer to this layer are random, and we take into account this large in degree, then we can estimate this feed-forward current as the sum of two components. A component that is not selective, so it is independent of the stimulus, and this is of order jk, and a component that is selective, so it depends on the stimulus, but is much smaller than the non-selective component, of order j square root of k. So in other words, if you take random projection, the inputs that this neuron receives will weakly depend on the stimulus itself. Okay? So it's not trivial to develop selectivity in this situation. Okay, so of course people have thought about it, and I show you three different solutions that have been proposed. So one solution is that is using feed-forward model. That basically says, okay, the projection from here to here are not random, but they are structured. So for instance, a neuron here will receive inputs only from a specific subset of neurons in the input layer. And this is what is done, for instance, in the Hubel and Wiesel model of simple cells in B1. Okay, and then the other assumption in this model is that synapses are weak, which means that they are of order 1 over k. So if you make this assumption, then the feed-forward input here is of order 1 and selective. Okay, then an alternative model relies on recurrent interaction. In these models, now neurons in this layer are not randomly connected, but connectivity is typically assumed to be like-to-like. Neurons with similar response property are more strongly connected, and this allows the network to amplify weak selectivity in the inputs. And if you take again synapses weak of order 1 over k, then you get that the network can show selectivity. Okay, then a third class of model. Doesn't make any assumption about structuring the connectivity. These are completely random. But synapses are assumed to be strong of order 1 or square root of k. And the network is in what is called inhibition-dominated regime. So in this model, what you have is that the non-selective component of the input is large, but is cancelled by recurrent inhibition. And the selective component remains in of order 1 and can produce selectivity. Okay, so this is just the overview that I wanted to give you. I showed you three different models that try to explain the same phenomenon, that is feature selectivity, relying on different assumptions about connections in the circuit. And so we try to, now, as I'll show you in a slide, now we have a lot of information about connectivity in the circuits, and so we try to use that to constrain these mechanistic models. Okay, so this data is what I call connectomes that are a reconstruction of the wiring diagram of neural circuits with synaptic resolution. I'll just spend a minute to show you how this data is obtained and what it contains. So a way to obtain these connectomes is through electron microscopy. One starts with a piece of brain tissue. Then this tissue is sliced in really thin slices. Each slice is imaged, and then the images are stuck together. And you have a visualization of the 3D volume. And then in this volume, you have the problem of reconstructing what's inside, and this is the segmentation problem. So the problem is the following. So you have this image, and you see that here you have sections of, for instance, an axon. you would like to track that section over different images, so to reconstruct this 3D structure. Okay? This is shown here with a moving. For instance, we focus on this piece of neuron here. You follow it into consecutive slices. And you keep doing it for the old volume. And you see that in this case, you get a dendrite making a synapse with passing by axon. This is a small piece of tissue. People can reconstruct entire neurons. And now it's an exciting time in the field because we start to have a reconstruction of entire brains. So the first one that became available was obtained in the 80s, and it's the reconstruction of the full C. elegans brain. This is a small brain, it contains only 300 neurons in this 1 millimeter long worm. And this reconstruction was done entirely by hand, and it took more than a decade to be made. Now, with artificial intelligence, this process of segmentation can be automatized. And over the last few years, we had the release of the reconstruction of the entire fry brain. And so I'm interested in cortex, and luckily enough, a few years ago, this Micron's collaboration released the reconstruction of 1 millimeter cube of mouse visual cortex. So this is a substantial fraction of the old neural circuit here. And of the same neurons, they also did in vivo calcium imaging while the animal was looking at natural images. So from this specific circuit, we have activity in vivo from thousands of neurons and connectivity between them. Okay, so we try to use that to constrain the models that I talked about earlier. So in the lab, we're interested in using this data to study complex computation and transformation of selectivity across the visual hierarchy in the mouse. Today, I will talk about the first step that we made in this direction that focus on a simple problem. So the problem is simpler in two ways. So first, we focused on a subset of the old circuit. We focus on layer 2-3 in V1 in response to input from its primary driver, that is layer 4. So we have a 2-layer network that we can model with the type of model that I described here. And then the second simplification is that we focus on selectivity to simple stimuli, in particular to oriented gratings. Okay? So these are just two example cells. That's the fire rate of a neuron as a function of the stimulus orientation in layer 2-3, and this is in layer 4. In both layers, we have orientation tuning. So we characterize that through an index that is called circular variance. It's defined here. It measures how big this modulation is. And this is the distribution of the circular variance in layer 4 neurons, and this is in layer 2-3 neurons. So the main question that we ask them is how can we explain these responses in layer 2-3 in response to inputs from layer 4 using the connectome to guide models. And going back to the previous work that I discussed, two questions that we're interested in are what are the contribution of feed-forward inputs from layer 4 and recurrent inputs within layer 2-3, and what is the role of structure in the connection versus randomness. Okay. So now, what exactly do we have? So this is our model adapted to this network. So it's a system of equation governing the activity of excitatory inhibitory cells in layer 2-3. And from the data, we have information of the volume of synapses that is proportional to the strength of interactions. So up to a proportionality constant, we have these connectivity matrices here. And the other thing that we have is that we, through calcium imaging, we know the activity in vivo of some of the excitatory cells in the network that are these terms. Then there are things that we don't have. In particular, the reconstruction is not perfect, so we don't have all the entries in the connectivity matrices. Not all the neurons are imaged. In particular, we don't have any information about inhibitory cells. And we don't have anything about the single neuron properties, like the time constant or the single neural transfer function. Okay, so now the challenge becomes how can we use this partial information to constrain these models? And we developed two alternative approaches to use this data. First, we use a plug-and-play approach of the connector. And the idea here is to look at observables that are less dependent on quantity, not directly constrained by data, in particular the synaptic currents. And with this approach, we show that recurrent interactions within layer 2-3 are the primary driver of selectivity. Then this is not okay. Then in the second, with a second approach, we try to study the statistics of neural responses. And here we use the data to constrain the recurrent neural network model and infer quantities that we don't know by fitting the model to the data and then doing the serial experiment on the fitted model. And in this model, what we find is that randomness in connectivity plays a major role over structure in shaping the selectivity. Okay, let me walk you through some more details of this. So for the first plug-and-play approach, so if you assume that the system is stationary, we have that the firing rate of a neuron as a function of the stimulus orientation is given by the transfer function of its synaptic currents. So this transfer function, we don't know it, but we know it's monotonic. Okay? So this already gives, we can use this expression to probe what are the role of the different currents in shaping selectivity. We can estimate that directly from the connector. This is how it works. So we take a neuron, this is its reconstruction, and this is its tuning curve. And then of this neuron, we can query the connector and look at what are its presynaptic partners. These are those from this specific neuron. And for each one of these cells, we can estimate the individual contribution that they give, so this term here, the firing rate of the neuron, and multiplying it by the synaptic strength. The example inputs produced by these inputs. Then we sum them, and we get these synaptic currents. So the total excitatory synaptic current is here in gray. And you see, depending on the angle, it's peaked at an angle that matches the preferred orientation of the postsynaptic cell, which is what you expect. Equation here. And then we can look at the contribution that come from recurrent interaction that are here in red, and the contribution that come from the feed-forward inputs that are here in yellow. And what you see is that recurrent interaction are stronger and more selective than feed-forward inputs. This is an example cell, but we see the same thing when we look at across the population. So this is the average synaptic current, and we have that it is larger than the layer 2-3 recurrent synaptic input is larger and more selective than layer 4. So what we find overall is that recurrent interaction seems to dominate over feed-forward input in shaping selectivity. Okay, so then going for the second approach, as I mentioned, we wanted to use the data to constrain the recurrent neural network model, which then we do experiments in silicon. And the properties that we observe, that we focus on, are those that are summarized here. So we have a distribution of rates, the average tuning curve of neurons, distribution of circular bias that I showed earlier. And something that will become important in a second is the fact that we have structure in the connectivity. So this is the probability of connection between two neurons as a function of their response property. And what we observe, consistent with previous data, is that neurons with similar response property are more strongly connected. And so what we do is to start from our models. We use information from the connectome to generate random matrices that follows the statistics of the connectivity that we extract from data. And then we infer the missing parameters with simulation-based inference. It is a technique that allows us to find a set of unknown quantities that best captures the data. So this works well. Now the continuous line are the best fit of the model. We can recapitulate all these observables. And we can also explain things that are not directly constrained from the data. For instance, the fact that inhibitory neurons are more active and less selective than excitatory cells. Okay, now in this model, we can do things that we cannot do in the data, in the animal. In particular, we can probe what is the role of structure. And structure in the model enters through this like-to-like connectivity. And in the model, we can disrupt it by just sharpening the connectivity in the network. so this is what we did. We take the simulation that describes the data. This is its circular variance, so selective neurons are. Then we shuffle the connectivity, so we disrupt structure, and this is what the circular variance looks like after that. So basically, we see that structure in this model plays a minor role in shaping the selectivity because most of it is already present when you don't have any structure in the connection. This is a single example, but then we see that over several simulations that provide a good description of the data. Okay, so I'm actually done. So just to summarize, we set out to study what are the mechanisms of feature selectivity using connectors to constraint models. We focus on a specific question that is how selectivity layer 2-3 emerges from layer 4 inputs for oriented stimuli. And we find that recurrent interaction seems to be the dominant contributor, and randomness in connectivity plays a major role over structure in shaping selectivity. Okay, if I conclude, thank you for your attention.
So I remember some years ago there was a similar discussion about whether it was coming from LGN, whether it was LGN that was triggered the orientation selectivity in V1. And David Furster did some beautiful experiments cooling these areas. Mm-hmm. So if you cool V1, you destroy the recurrent connectivity, but you're still receiving input from LGN. And if you cool LGN, then you can see only what arises is only due to the recurrent activity in V1. And I was wondering, and that resolved that issue, but I was wondering whether one could do something similar at the layer level. Do you know if that experiment is possible?
I know at the layer.
I'm a theory, so I really don't know.
Yeah, no, at the layer level, no, I don't know. Something similar has been done in mice for layer 4 by optogenetically silencing cortex, but then again, it was layer 4 that is the primary target of the LGN. Then from layer to layer, it's not been done as far as I know. Okay. I know that there are, so from the genetical point of view, layer 2-3 and layer 4 neurons are different. So you could, in principle, target.
Oh, maybe an optogenetics experiment to silence. Yeah.
But I don't think this has been done.
Okay. Thank you.
I think very exciting work, and it's very good that you're sort of taking into account this new data that is coming with micron project. I'm curious about how, because what you get from the electron microscopy is, I mean, essentially anatomical connectivity. And but the anatomical connectivity is not the same as, doesn't directly give you the strength of the synapses.
Yeah, so we rely on, there is one paper that I know that showed that the size of the synapse is proportional to the strength of the interaction. So the size is also part of the data. For every synapse, like tell you how big it is. And so basically this is what we assume in the model. Basically, the connectome gives you after proportionality constant the full connectivity matrix. And when we analyze single neural responses, that proportionality constant doesn't matter because it multiplies all the inputs in the same way. So it affects the selectivity of the inputs. Then for recurrent dynamics, that matters a lot. And what we do is to infer it from the functional data.
Also a question. So I was wondering about the contrast between the highly detailed resolution of the morphology and then the fact of the morphological data, and then the fact that you have just two populations. So I was wondering, I mean, if you're selecting two populations out of the data, and or you're clustering like all excitatory or pyramidal and neuro and inhibitory subpopulations into RE and RI.
Yeah, so they have a classification of cells based on the morphology. Yeah, but this is done at different level. So the more constrained description is just excitatory and inhibitory, and we took that. Okay. Then no, it's possible to go beyond that.
Maybe not necessary.
I don't know. I guess it depends on the question. because it's clear that different inhibitory cell types, in particular, different patterns of connectivity with excited cells. So it would be interesting to study that. It's just we've not done it yet.
Okay. Are there other questions? No, let's just proceed.